Good morning, everyone, and welcome to this morning's Pediatric Grand Rounds. Before I introduce our speaker, I'll read our land acknowledgement. Ottawa is built on unceded Algonquin and Anishinaabe territory. The peoples of the Algonquin and Anishinaabe Nation have lived on this territory for millennia, and we honor them in this land. Their culture and presence have nurtured and continue to nurture this land. CHEO also honors all First Nations, Inuit, and Métis peoples, and their valuable past, present, and future contributions to this land. I'd ask everyone to take a moment to reflect on that. So it's a privilege and a pleasure for me to introduce today's Grand Round speaker, Dr. Hans Lochmuller. And if I read all of Hans's bio, that would be the end of Grand Rounds because he has had a very illustrious career. Hans is a senior scientist at both the CHEO Research Institute and the Ottawa Hospital Research Institute and the Brain and Mind Research Institute at the University of Ottawa. He is a professor of neurology and a Tier 1 Canada Research Chair in Neuromuscular Genomics and Health, and probably really one of the world's foremost authorities on neuromuscular disease. And Hans really epitomizes, I'm not reading from this here, Hans, I'm going off script. Hans really epitomizes the clinician scientist taking work from bench to bedside. You'll hear about some of his preclinical models. Today he's going to focus on congenital myasthenic syndromes, but he studies neuromuscular diseases in the lab using mice, using zebrafish, and then takes these discoveries to the clinic where he looks after children at CHEO and adults at the Ottawa Hospital. Hans trained as a neurologist in Germany and in Montreal. He leads a large research group here at CHEO-RI and focuses on molecular therapies of neuromuscular disorders, molecular pathogenesis of muscle and neuromuscular junction disorders, and neurogenic and translational research, including genomic approaches for diagnosis and gene discovery in neuromuscular and rare diseases. And with that, Hans, thank you for being here, and we look forward to hearing your presentation.
Well, thank you so much, Jason, for this very kind introduction. And it's a special pleasure to be here back for Grand Rounds. Last time I talked about this topic was pre-pandemic, so it's a while. And of course, I've talked about this topic elsewhere, but it's nice to bring it back home where actually most of this work is being done that I'm presenting to you today.
Just hover down the knife straight down. So you just can't see if you have them. Yep.
All right. So hope everybody's hearing me now as well online. Hello, everyone. And before I start, I just want to show you my disclosures. So as Dr. Berman explained, I'm doing a number of clinical trials, both here at CHEO as well as OHRI. And these are with various pharmaceutical companies. The ones that are relevant for this talk are the ones highlighted in yellow, Argenx and Neurotune, and I will come to that when we get there.
So you're buying beer the next time they go out.
So, objectives of my talk, first of all, how to diagnose and treat clinical congenital myasthenic syndromes. So that's the more clinical part. And while congenital myasthenic syndromes are very rare and not everyone may have encountered them in clinic, I think it's particularly important to recognize them because for most of them there is already a treatment available. And secondly, how to investigate neuromuscular transmission disorders in model systems. So the neuromuscular junction is probably the best understood synapse in the human body. So we learned a lot from both investigating animal models and humans, and this is relevant for this rare disease, but is also relevant for many other situations. Just think about how it is possible that you sort of have a thought about going out the door when you hear me talking and then signal this to your muscle. So how does that work? Well, this is what we call neuromuscular transmission, and there's several receptor and pathways that we now understand much better and that are in fact defect in these genetic conditions. So the name congenital myasthenic syndrome is a little bit misleading. Because not all of these children present directly at birth, but the majority, around 80%, present within the first 3 years of life. And how does it look like? So I brought a video here from one of the children that's affected by congenital myasthenia. So what you see, the child has some facial weakness, some droopy eyelids, ptosis. She can walk on the flat, but at that time she's unable to walk stairs. If she's asked to sit down on the floor, that works well. Down is always easier than up. But then if she's asked to stand up, then she presents what we call a Gowers maneuver. So she pushes herself up with her hands on her thighs, and this is because she has limb girdle weakness. And this is true not only for the legs, for the lower limbs, but it's also true for the upper limbs. If she's asked to lift her arms, she has a hard time doing that. And if she's asked to do this second or third time, then it becomes almost impossible. So what you see here is both the weakness, proximal weakness, then the ptosis, plus the fatigability. So it gets worse at repetition or as the day goes on, etc. And these are hallmarks of myasthenic syndromes generally. The more common disease is, of course, myasthenia gravis that many of you will know about, which is an autoimmune disorder. In this case, there's no antibodies. This is a genetic disorder. And we find out, and I'll come to that in more detail, that this child has recessive mutations in the DOX7 gene that explains her congenital myasthenic syndrome. So how do these conditions present generally? So the presentation is actually quite variable. It can start right at birth and present as a floppy baby, plus minus arthrogryposis, maybe look like spinal muscular atrophy. It can present more in young adults with ptosis and ophthalmoplegia and maybe mistaken as a mitochondrial disorder. Or it can present with muscle atrophy, loss of ambulation, scoliosis, respiratory distress in adults, and look more like a muscular dystrophy. So it's a fairly broad phenotype, and because the vast majority of these conditions is recessive, nowadays in our small families, it's often just a single case, a sporadic case, and that's why it's rather difficult at times to diagnose it. So what helps to secure the diagnosis? Starting, of course, with the clinical suspicion, meaning if a kid is weak and not only gets worse at times, but also gets better at times, more than what you expect from a progressive condition like muscular dystrophy, then that's a point to think maybe it's not a muscular dystrophy or spinal muscular atrophy or some of the more common neuromuscular conditions. Maybe it is affecting neuromuscular junctions. And then we have a test, clinical neurophysiology, which is, for instance, done here at CHEO by Dr. Macmillan, who would do a repetitive stimulation, meaning applying an impulse, an electric impulse over the nerve repeatedly at 3 per second, and then the muscle action potential would get lower from one stimulus to the next, what you see in the right upper part of the image. So this is quite indicative of a failure of neuromuscular transmission if you see that decrement. In some cases, of course, people have suspected first is this a myopathy, a muscular dystrophy, and they go on to do a muscle biopsy. And in that case, the pathology report may come back and say, well, there isn't much wrong with this muscle. It's at most a mild myopathic tissue with maybe some rounded fibers, some internalized nuclei, but it's not muscular dystrophy. And this is the next point in time where, as a clinician, one should think, well, maybe this is something else. Maybe it's not the muscle that's affected, it's possibly neuromuscular transmission.
Sometimes it stops there and nobody investigates anymore.
Yeah, it does. Yeah, and it's often that it's then told to be some unspecific weakness, myopathy phenotype, and it's not being looked into detail. Nowadays, of course, much easier because we have all these tools now for molecular genetic testing, panels, exomes, genomes, and that's what obviously brings up the diagnosis, sometimes quite surprisingly. So people did not think about congenital myasthenia, and then they see, well, this gene finding really tells them what it is. Many of the patients do respond favorably to certain types of drugs like esterase inhibitors, and if that is a very clear response, then that also helps to secure the diagnosis. Muscle MRI does also help to differentiate from these other conditions that I mentioned, but the mainstay really, and the way we secure nowadays the diagnosis in around 70 to 80% of the kids with congenital myasthenia is genetic testing. So what do we find with genetic testing? Well, it's not just one gene that causes congenital myasthenia. That would be a bit too easy. By now we know about 35 different genes, and they are distributed in terms of what proteins they encode for to the nerve. So these we call presynaptic, to the muscles, these we call postsynaptic, or to the synaptic cleft. Some of the genes actually do not correspond to a protein that is exclusively expressed at the neuromuscular junction, such as glycosylation genes. And there we have a harder time to explain why these genes specifically cause congenital myasthenia, but we'll come to one of these examples. But there's some sort of pretty obvious candidates like the acetylcholine receptor, and that's composed of 4 different subunits. So each of these subunits, if there are recessive mutations, causes congenital myasthenia, for instance. So that's one of the pathways that's pretty obviously involved. But there's a second pathway also depicted on this picture that I will come to later, which is the acquin LRP-MuSK pathway, which is important for forming and maintenance of the synapse. So it's not directly involved in the signaling to make the muscle contract. It's rather sort of a more long-term effect on synapses. So I'm not going to talk about all the 35 genes, but I'll just pick out 2. And the reason for picking out those 2 genes is because they are the most common ones, and they have particular clinical presentations or phenotypes that you can keep in mind, and that will sort of guide you towards maybe even a genetic diagnosis, or to suspect it at least. So the most common form of congenital myasthenia is recessive mutations in the epsilon subunit gene, the CHRNE gene. And the onset is usually in the first 3 years of life. The symptoms are ptosis, weakness, and occasionally respiratory insufficiency. Mestinon, so esterase inhibitors, do work in these children. Sometimes they need a second medication as an add-on. But one of the symptoms that is very peculiar and should be tested is that these patients do almost always present with fixed ophthalmoparesis. So if you ask them to follow your finger, your reflex hammer, with their eyes, they can't actually move their eyes. They will just move their head to follow your guide. And if you ask them, is there something wrong with your eyes or with your vision, they will say no. Everything is absolutely fine with my eyes, with my vision. I see clearly. I don't have problems seeing, right? So this is a sign that you actually need to test. And they have got used to that from early childhood on. So they don't have double vision like patients with myasthenia gravis would have. And they compensate with moving their head. So an important sign, which is found in those patients that have receptor deficiency and not in some other forms of congenital myasthenia. So that's why it's quite an important sign. Of course, it's also found in mitochondrial disease, so it's not proving that this is congenital myasthenia, but of course, in combination with everything else that I said, it's very helpful. The other gene which presents a bit different here is the DOX7 gene, and this is causing a congenital myasthenic syndrome of the limb girdle type. So these patients may also have ptosis, but they don't have the ophthalmoparesis like you see in the young man. He can actually move his eyes quite well. But their symptoms are mostly related to muscle weakness in the proximal muscles of the legs and arms, like you saw actually in the video of the girl in the beginning. And what's also important, esterase inhibitors are really not working in this condition. So if you try it on these patients, they come off very quickly. So they are ineffective. We use other drugs, and I'll come to that in a second. So what just these two examples may have shown you is that there's different presentations of that congenital myasthenic syndromes depending on the gene, and so are the treatments. So there are a larger group of patients with different genes, not just the receptor subunit genes, that do respond well to pyridostigmine, to mestinon, and then that's the drug that you should start treating them with and dose them up to like 5 to 10 milligrams per kilogram per day, to very high doses actually. And then if that's not sufficient, then you may combine this with 3,4-D-immunopyridine or salbutamol. And then there's this other group that clearly does not respond well to pyridostigmine, or where you can actually make things worse. And this includes DOX7, as I just shown you. This also includes CoQ, which is actually causing a defect in the esterase to start with. So if you block the esterase further with mestinon, you're making things clearly worse. And this is so-called slow channel syndromes. And for the majority, the starting drug is nowadays salbutamol. Sometimes in the past we used ephedrine, which is a beta-2 sympathomimetic drug. And that seems to have a direct effect on the synapse. And we've done some work that I don't present today, but there's still ongoing work in the lab to understand that pathway better. So this is the same girl again that I showed you with the first video. She's now diagnosed with her DOX7 congenital myasthenia, and she's on salbutamol for about a year. And she's now asked to get up from the floor. Oops. Let's try to get that video running. Here we go. So now she can get up from the floor without a Gauss maneuver. She lifts her arms quite well, and you also see that her facial appearance has really changed. And in the next video, she's asked to walk stairs. She couldn't do that at all at the time when I showed you the first video. And here, well, she's holding on briefly to the railing. She's not exactly running up the stairs like some other kids would, but she's able to walk at least one flight of stairs, which obviously makes it much easier for her to attend school and everyday life, etc.
She's also gained weight a bit.
Probably.
Is that what happens with the therapy?
Yeah, I think generally a healthier appearance, I would say, yeah. They don't always have sort of feeding difficulties with DOX7. That's more common in some of the other subtypes. But of course, now that her muscles actually respond better, she is able to carry a little more weight, I guess. So I switch gears now a little bit and come to what we do in terms of figuring out those 20-30% where we do not understand the genetic basis. So those are the ones where we've done panel testing or exomes and did not come up with a clear answer a mutation in one of the 35 genes that we know about. So in those, obviously, we do more sequencing, exomes and genomes, and then do a lot of reanalysis as well of existing exomes and genomes. And the sort of workhorse that we use for this is the RD-Connect genome phenome analysis platform. So this is a big genomic database, basically, over in Barcelona, where lots of different people have deposited not only exomes and genomes of their patients with neuromuscular and other rare disease, but also phenotypes. So that makes it different from just a variant database. So there's phenotypes associated in terms of HPO terms, and that helps you actually make sense of the variants and the genes that you identify. So using that, we have investigated, and that was work that Kieran did. He's now a postdoc in my lab, and he was previously based in Bangalore in India. So he, together with his boss at the time, Dr. Nalini, saw this pair of brothers that clearly presented with fatigable weakness, with ptosis. They were from a consanguineous family, and both had also some additional central nervous system symptoms like seizures, which occasionally occur in congenital myasthenia, particularly with some of the presynaptic genes, because they may also play a role in the brain. But they were both investigated and had a decrement at repetitive stimulation, so it was clearly felt they have congenital myasthenia. And again, here are some of the still pictures that show the difficulties with walking. I'll show you in the next video, actually, the walking. So this is one of the young men, and he is able to walk. He's not very quick. He needs to sort of help a little bit. He's a bit insecure. And then, as my colleagues suspected that they do have congenital myasthenia, they put him on salbutamol. And this is after a couple of months of salbutamol. So again, he's not running, but he doesn't need help. He walks much better. He walks further. So a positive effect, I would say, of the salbutamol clinically. But as we didn't find any mutations in the known genes, we went on with some more testing, and a muscle biopsy came in this case, and that showed alterations that we did not expect, actually. It showed both COX negative fibers as well as ragged red fibers, which are hallmarks of mitochondrial disease. So clearly there's a mitochondrial component to this condition. And by analyzing the exome of both of these brothers, we identified a homozygous variant in the TEFM gene, which is a mitochondrial gene and codes for a protein that helps with the elongation of mitochondrial transcripts. So that's where the name comes from, transcription elongation factor in mitochondria. So with that, well, that's interesting. It was not known as a disease gene at this point, but obviously it has an important role in mitochondria. But how does that then lead to congenital myasthenia? I mean, there's overlap in terms of the symptomatology between mitochondrial disease and congenital myasthenia, like the ptosis, like fatigability, etc. But usually you would expect a more sort of multi-systemic presentation of a mitochondrial disease affecting the heart and the brain and all sorts of other important functions. So when we then actually looked for other patients with mutations in this gene, we did actually come up with some patients that have more classical mitochondrial disease. So kids that died early, that had brain abnormalities, that had cardiac or liver failure, and they all had similar mutations in the TFM gene like our patients with the congenital myasthenia. So what we did then next, and this was done mostly by Emily O'Connor, postdoc in the lab at the time, we used one of our model systems, zebrafish, and we knocked down the TFM gene in zebrafish embryos. So what the advantage in zebrafish is that you can look at many animals at the same time, and also they develop very quickly. So from the fertilized egg to swimming around in a tank, that's only 48 hours. So if we knock down the TFM gene in zebrafish embryos, what do we find? Well, looking at the neuromuscular junctions first, so the upper row is the control, where in red is the receptor stained with alpha-Bunger toxin, and green is the nerve. And in the overlay in yellow on the left-hand side, you see the neuromuscular junctions. And this is how they look in the zebrafish body, which is sort of mostly muscle, actually. In the lower part, this is the knockdown zebrafish. And again, red is the receptor, green is the nerve, and yellow is the overlay. And you see this is a lot less sort of regular, and there's less yellow, less overlay. So there's disrupted neuromuscular junctions in these zebrafish embryos. And when we looked at these zebrafish embryos at 48 hours, when they swim around in the tank, then we observed the difference again between those controls in blue and the knockdown zebrafish. So zebrafish do swim more when the light is off. That's when they get active. And then they stop swimming or swim less when the light is on. So what you see here on the left side is light on, light off. And the more often the light is off, so the more often they start swimming, the more you see the difference actually between the knockdown zebrafish and the control zebrafish embryos. So meaning there is this weakness and fatigability that's expressing itself in that mobility phenotype. So what we found is that TFM is a novel mitochondrial disease gene, and Dr. Minczuk's and Dr. Horvath's lab in Cambridge did a lot more very sophisticated work to actually prove that these longer transcripts that particularly rely on the TFM protein are more affected than the short mitochondrial transcripts. The disease is variable and early onset and often involves other organ systems, but it can lead to a more pure congenital myasthenic syndrome. So it's an interesting condition, but it shows that mitochondria are actually quite important for neuromuscular transmission. And in the meantime, we have actually identified additional mitochondrial genes that also cause neuromuscular transmission defects. I'm coming now to this other receptor system that I mentioned briefly, the agrin LRP4 MuSK-DOX7 complex. So that's a bit of a mouthful here. And I've got this sort of colorful cartoon from my colleague Ruth Herbst in Vienna, who basically spent all her career figuring out what this complex is doing. So I'll walk you through it in a 1-minute cartoon. So pay attention. So the green and yellow molecule, that's the LRP4. The blue one is MuSK. And there's each two of them sort of build a receptor, a heterodimeric receptor. So if agrin comes in, that's secreted from the nerve ending, it binds to LRP4. That leads to a change in the conformation of the receptor. That leads to phosphorylation of MuSK and activation of MuSK. So that's a critical step here. That recruits DOX7, the gene or the protein that we talked about before. That leads to further phosphorylation. And a signaling pathway leads to phosphorylation of the acetylcholine receptors and to clustering of the acetylcholine receptors, amongst other things, to maintain the synapse. So you see here how DOX7, while it's not directly involved in the signaling with acetylcholine plays an important role here in this system, and that's why it's causing congenital myasthenia. So, of course, if one of these molecules is defect, you get congenital myasthenia. It's not a surprise. That's true also for agrin, LP4, and MuSK. But what can we do about it? Well, the sort of simple answer is, well, if there's a defect, if DOX7 is missing, well, let's bring some more DOX7 in, do gene therapy. And that does actually work, at least in mice. So if you replace DOX7, bring it in with an AAV, you can cure your DOX7 mice. DOX7 mice, by the way, are a very severe phenotype. They die at 7 days after birth. But you can, if you inject your AAV early enough, you can rescue them. But AAV gene transfer, as you all know, works in certain conditions, but it has also certain disadvantages and risks. So there was a very clever thought by Dr. Burden, who is a researcher in New York, who said, well, if we don't need to replace DOX7, we just need to replace the function of DOX7. And maybe there's other ways to activate MuSK and phosphorylate MuSK. And this is what I'm going to talk to you for pretty much the rest of this seminar. So he developed so-called MuSK agonists. So these are antibody-like molecules that bind to the neuromuscular junction from outside. So it's an antibody treatment in a way, not a gene therapy. So if you inject these MuSK agonists into the DOX7 mice, which, as I said, usually would die at 7 days of age, which is depicted here with the red arrow on the right side. So the little gray symbols are the mice that stop at 10 days in this case. So they all die. If you inject those mice just after birth, and then again at 18 days and at 38 days with these MuSK agonist antibodies, they do not only survive, but they gain weight, and they are pretty much indistinguishable from their wild-type littermates. So it's a dramatic reversal of the phenotype, I would say. So based on these results, that at least for mice are very promising, we and others have teamed up with a pharmaceutical company called Argenx and started a clinical trial, a first-in-human trial for congenital myasthenia with a similar compound, with a MuSK agonist. So this is a trial where 15 patients have been included worldwide in about 10 centers, and our center was the first one to recruit and treat a patient with this new compound. These were all adults for safety reasons, but hopefully it will come soon to children as well. The design is quite interesting. So while in most sort of early trials or phase 2 trials, you would have one group on the lowest dose of your new medication and then slowly go to the next dose for the next group of patients. This was actually a dose escalation in each patient. So the first infusion that the patient got was at 1 milligram, which was the lowest dose. And then every 2 weeks, you basically increase the dose of the MuSK agonist up to 10 milligrams per kilogram. And also, it was at intervals that were, you would at least assume that both the antibody as well as the effect of the antibody may be accumulating over time. So all these patients have now been finished with their treatment, and we're currently waiting actually in their follow-up how long the effect will last, because at some point, I guess all the antibody will be gone from the system and the phosphorylation might be lost, and then we would expect them to get worse.
There was an effect?
Well, there was a clear effect. I mean, I'm blinded. I don't officially know who got the placebo and who got the drug, but I would say there was a very clear effect. So as I said, these were adult patients for now. They were on the more severe end of the spectrum. So in fact, in my adult clinic, in a natural history study, I have seen 12 patients with DOX7 congenital myasthenia. Only 2 actually qualified for this particular trial. And that was basically on severity. And the main outcome measure that's being looked at is the QMG, which is an outcome measure where you count seconds, how long you can hold up your arms and your legs and your head, etc. And that goes into a score, and that's been an outcome measure that's been used in myasthenia gravis a lot. Not in congenital myasthenia, because there were no trials in congenital myasthenia. So in addition to obviously figuring out whether this new drug works or not, we do learn a lot about what sort of outcome measures work in congenital myasthenia for future trials. And of course, we do a lot of other tests as well in these patients. So I'm expecting those results to come up later in the summer, and hopefully then additional trials will start, including trials in children here at CHEO. The next question that we had is, well, if that works in DOX7, at least in DOX7 mice, could it potentially work in other forms of congenital myasthenia? Because I've told you in the beginning, there's like 35 different genes, and some genes we have fairly good therapies, but other genes we don't. So we picked 2 that we felt would benefit from a better treatment, and where we had actually good mouse models here. So one is a defect in agrin itself. So agrin, as I've shown you before, is sort of part of that pathway. Obviously, that would make a lot of sense if you can replace that through a MASC agonist. And the other one is CoQ. Well, CoQ isn't such an obvious candidate to start with, other than that, well, there isn't a good treatment for CoQ congenital myasthenia, but it also binds to MuSK, and the function why it binds to MuSK and what the relation is not entirely clear. So Kelly and Ofosu and Ricardo and Sally did a lot of work on these mice to test MuSK agonist antibodies, whether they would work. And I just briefly walk you through that. So the top part of the slide is the agrin deficient mice, and the lower part is the CoQ deficient mice. Agrin deficient mice, so the mutant mice are the blue curve, and the wild type controls are the green curve. So agrin mice do not die as early as DOX7 mice, but most of them will die within the first 2 months of life. And that's what you see on the left side, right? So the blue curve goes down. So if you give those mice a MuSK agonist antibody treatment, then you get the red curve, which basically means they do survive like the wild type mice. So there's a clear survival benefit for these mice. And then on the right upper graph, you see grip strength. So the blue mice, the mutant mice, have less grip strength than the wild type mice, as you would expect. And if you treat them, then their grip strength at least is partially restored. In the lower graph, you see the CoQ mice, and there you see that, in fact, the blue, the mutant mice, and the green, the wild-type mice, they don't show any difference in terms of survival. So these CoQ mice, at least if they're treated well and you put food on the floor so that they get rigid, they do survive. So obviously then giving them a MuSK agonist antibody won't change that, hopefully. But on the right lower part, you see that they do have a defect in strength, so their grip strength is reduced. And if you give them a MuSK agonist antibody, then the red ones actually do not show an improvement. They stay where they were without the treatment. So that first sort of experiment meant, well, possibly agrin deficient mice do benefit, but CoQ deficient mice maybe not. Looking into this with a lot more detail, At the neuromuscular junction, similar to what I showed you in the zebrafish, we did similar stains now for the neuromuscular junction in the acumen in the CoQ deficient mice, and derived a number of parameters how healthy their neuromuscular junctions are. And this is shown on the right-hand side, both in terms of the nerve terminal parameter and the complexity of the neuromuscular junctions. And in the acumen mice on the top, you see that this is reduced in the mutant mice in blue, and it's restored with the MuSK agonist antibody. And on the lower right, in the CoQ deficient mice, again, it's reduced in the mutant mice in blue, but it's not restored with the antibody. So that goes hand in hand with what I've shown you on survival and grip strength. And finally, probably the most important one here is, well, is it actually the mechanism that I told you about? Is it MuSK phosphorylation that makes the difference? And here on top again, you see the acumen mice that have reduced MuSK phosphorylation on the Western blot in blue, and it's restored with the MuSK agonist antibody, while there isn't much of a restoration in the CoQ deficient mice, and in fact the deficit isn't quite as clear in the CoQ deficient mice. So maybe it is a different mechanism and we don't restore it with activating MuSK. So coming to my general summary, congenital myasthenic syndromes are clinically very variable and heterogeneous. There are some clues pointing towards the underlying genetic defect. The exact prevalence of CMS is unknown, but may be underestimated. The next generation sequencing continues to uncover new genetic causes of CMS and helps broadening the spectrum of inherited transmission disorders, and the detection of the genetic defect is important for the selection of the appropriate drug therapy. Modeling in cells and animals is critical to understand the role of disease genes, the impact of mutants, to identify pathways and therapeutic targets, and to test therapeutic approaches. And a little take-home message for clinicians, so particularly, of course, pediatric neurologists here have a high level of suspicion for CMS. It's underdiagnosed, so there are patients that did have symptoms in childhood and are only diagnosed later, even as adults. Use all types of genetic testing for diagnosis, from panels to research genomes, and if you're at the end of the road, what your public payer is sort of providing with, please come see us and we'll figure something out. Discuss patients with unclear diagnosis. They will be considered for gene discovery and other research projects, and offer CMS patients to sign up to registries. In Europe, that's the EuroMD registry, and here in Canada, it's the CNDR. So there is a specific module actually for patients with congenital myasthenia, because every center will only have a handful of patients with CMS to start with. So obviously, the more we centralize that information, the better we can understand these conditions, their genetic background, and also power trials for them. And what's probably most important for patients, there are clinical trials now that are moving from preclinical and to clinical, and I showed you the first example, but I think there will be more to come. And I thank everyone, particularly my team, on this picture, having fun skating on the Vito Canal earlier this year, and a lot of collaborators internationally. Thank you so much.
Thanks so much, Hans. The sad thing is this picture could be taken today, but that's a whole other story. And, as sort of promised in my introduction, Hans really showed us how beautifully you can move from the clinic to the lab, back to the clinic, and use preclinical models to inform diagnosis and novel therapies. So we have some time for questions. Hugh.
Yeah, thank you, Hans, for that excellent talk.
Sorry, and I'll just ask Hans, just repeat the question after for the people online in case they can't hear.
Yeah, no, thank you. So a comment and then a question. So the comment is, I I'm optimistic that we will pick up more patients now that the government of Ontario has approved clinical exomes for children less than 2 with neuromuscular symptoms. So I think hopefully that will help.
Okay. Dr. McMillan said that Ontario is now paying for exomes in children under 2 with neuromuscular disease, which is great.
Yeah, and then my question is, it's around access of treatments. So there's been challenges because we no longer have access to 3,4-DAP. It's been cut off since it's had narrow approval for an adult condition, LEMS, and salbutamol is also no longer manufactured as the pill. So the liquid is really troublesome because it has such a rapid onset of action if we give them like the little ampules. How do you deal with that?
Yeah. No, Dr. McMillan asked about access to treatments, which is particularly difficult for 3,4-DAP since it's now only approved for a narrow adult condition, as well as salbutamol, which is no longer available as the pill, only as a liquid, which is much harder to get right. So yes, that is a big problem, and the 2 drugs that are readily available, well, is methadone, obviously, and no problem here, thanks God. And then there is also, of course, fluoxetine, and there's a little bit of a warning here. So fluoxetine should really just be used for slow channel syndromes, so for the dominant form where you have prolonged opening of the acetylcholine receptor channels. All other congenital myasthenias will not get better with this drug, they will get worse. But because it's more readily available than the other drugs that we just talked about, doctors think, well, if I can't get drug 2 or 3, well, I go to drug 4. But that's wrong. It should really just preserved for slow channel syndromes, which is only about 5% of all congenital myasthenias, is only the dominant forms, really. So coming to the core of your question, 3,4-diaminopyridine now is a marketed drug, and therefore we can't even get access to the old unlicensed stuff anymore that we sort of usually got compounded by the hospital pharmacy. So there is at least some attempts to get this on a per patient basis through Ontario special access programs. So that's in the works, and of course there's actually 2 companies now in Canada that market this drug for this very rare conditions, and you can on a per patient basis discuss with them to get access to them, but it involves a lot of paperwork, so it's become a lot more bothersome. I did not need to take off a patient from their DAP so far, so I think it's got a lot harder, and it caused a lot of frustration that the old 3,4-DAP is no longer available, but I think we're not going back there. And we also don't see a way how for congenital myasthenias a company would start a clinical trial just for this drug. I think that's unrealistic. For the salbutamol, that's sort of an ongoing struggle. So there are companies in Europe that still produce it, and Ontario or Canada would allow, Health Canada would allow to import it, but then it's not commercially viable actually for this company to import it. So that's why it's not happening. There are still some compounding pharmacies that actually do it, which comes at a cost.
Make pill?
To make the pill?
Yeah.
And these compounding pharmacies, we have a few addresses that obviously are mostly here in the Ottawa-Gatineau region, so I don't know how it works in other places. But that comes at a bit of an extra cost for patients, right? So it's that's the trade-off. But I think that's still probably the better way than the syrup or the fluid from the puffers.
Thank you.
Thanks for that question. I would just add that this is part of an ongoing challenge for rare genetic disorders and access to drugs. And the end of this week, the Rare Kids Can meeting is here in Ottawa, and so I think it's important to remember, like through the through the strength of this national organization, the advocacy piece, which of course includes patients and families, which are really the strongest voices to government and pharma. Alex.
General audience, at the ballpark, how many congenital myasthenia families, patients do you see in the Ottawa area and the Captain Basin?
Yeah. So probably purely in the Ottawa area, we have about 10 families with congenital myasthenia. We do now, because the programs get known, draw patients from other provinces as well.
Right.
So probably any of our neuromuscular colleagues who have a question about a patient with congenital myasthenia that they can't answer themselves, they probably come to us and ask.
And you feel it's underdiagnosed. If you go into the general population, the huge UK biobank, and look for the mutations, is it lurking out there? How many more do you think there are out there?
Yeah, so Dr. McKenzie is asking about, are there more patients lurking out there? And if we go into genetic databases, can we estimate how many patients we don't diagnose, overlook, or aren't severe enough or whatever? So yes, we did that and looked into Genomat to start with. So, and there's a heterozygous frequency associated with all these variants. And then with a formula, you can actually calculate how many homozygotes for this mutation you would expect in the population.
Hardy-Weinberg.
Exactly, Hardy-Weinberg and some other calculations that we did together actually with my postdoc Rachel Thompson and Dr. Perkins. And we did this for some similar conditions as well. So it appears that there's probably at least threefold more patients out there than we do diagnose. And this is in a way reflected in epidemiology literature that looks at congenital myasthenia from different parts of the world. So the estimates range between like 1 in a million to 1 in 100,000. So there's like a factor 10 in these. And of course, it could be ascertainment bias, etc., or differences in the population. That's entirely possible. But I do think that there's quite a few patients out there that we don't diagnose at this time.
I've got one more if there's no others.
Let me just see if there's any online. I don't see any questions online, so please go ahead.
Between siblings sometimes suggestive of genetic modifiers.
Yeah, a question by Dr. McKenzie. Phenotypic splitting differences between siblings that would sort of indicate a modifier. Yes, that there is. So there's differences. And so the most common mutation in congenital myasthenia is a specific mutation in the epsilon subunit. It's one that originated in the Roma population that emigrated out of India about 1,000 years ago and then spread across Eastern Europe. So this specific mutation on this founder haplotype is the most frequent mutation. And we did, together with colleagues in Bulgaria, assemble a cohort of about 100 patients with this mutation homozygously, which is obviously not easy for a very rare disease, and then looked at the sort of 10% most severe and the 10% least severe patients and compared them in sort of a multi-omic approach with genomics and RNA-seq, etc. And it's a paper we published recently in Nature Communication. It did actually show that there are modifiers, but it's not the same modifier in every family. It's like a different modifier pretty much in every family, which sort of is often related to a heterozygous mutation in one of these other congenital myasthenia related genes or other genes that have something to do with neuromuscular transmission. So it's sometimes worth, I guess, if you see a patient that is a lot more severe than you would expect with this gene or this mutation, to see is there a second hit somehow. I think that's what I would take away from that clinically.
One quick question just related to your question, Alex. I think these patients hide in 3 different phenotypes. So I think one are SIDS, so these patients that will just die. often in the context, especially the CHEO mutations that have a mild respiratory infection. I think the second is the myopathy phenotype that there's no genetic diagnosis for. And then I think the third, which hopefully is more rare, are the mistaken for an autoimmune myasthenia gravis that's refractory to immunosuppressant therapies. So they hide in different places, and some of them can't be detected because if you die of SIDS, you're gone.
Yeah, over to do genetic testing on them all.
Yeah, so Dr. McMillan commented that they are hiding congenital myasthenia in 3 different places: in sudden infant death syndrome, in the unspecific myopathies, and then in possibly seronegative myasthenia gravis. So we looked at number 1 and number 3, in fact. So the SIDS, so that's mainly the CHAT and rhabsin mutations, some presynaptic genes, and it's difficult to get hold of larger number of DNAs from kids that died from SIDS, right? Because before they're not symptomatic and you don't have DNA samples from them, or they don't get sequenced. We didn't get an awful lot of these patients, but you're right, in families where we then maybe diagnose the second kid with CHAT or rhabsin, there's often a kid that died of SIDS. So I agree with you, but we couldn't prove it. The third one, the seronegative myasthenia gravis, we've done that together with a colleague in Brazil, Eduardo Esteban, who had a larger cohort of seronegative adults with myasthenia gravis. And out of those, about 10% would have actually a causative mutation in a congenital myasthenia gene, which doesn't sound very much to start with, but given that myasthenia gravis is so much more common, and therefore seronegative myasthenia is also more common than congenital myasthenia, there's quite a few hiding out there as well. So it's worthwhile if people have patients with seronegative myasthenia that do not respond well to sort of the standard treatment with steroids or immunosuppressive drugs to rethink and do a panel or an exome on them.
Hans, in our last couple of minutes, maybe I can ask you a more general question. You've highlighted the fact that you've got many international collaborations, and so I just wonder, for people listening both here and online, how do you make those kinds of connections when you work in rare pediatric disorders? How do you connect with those various colleagues so that you can get the sample sizes that you need to do this work?
Yeah, I mean, one thing that obviously helped me personally is that I worked for many years in Europe.
Pediatrics in Europe.
and that's how I build up collaborations.
But you mentioned Brazil and other places that you haven't worked directly. So.
And then it's often when on a sort of per patient or family basis, colleagues contact me and have a question about their patient or their patients, that we find out maybe there's an interesting research question, and then we obviously need to look for the money to get some funding or get a fellow over to do the work, etc. But it's often starting with a clinical question, to be honest. And then, of course, some of the bigger projects that we've done in Europe, like RD Connect or Neuromics or Solv4D, they did help make these connections and build these infrastructures. And we still use them, but now actually the good news is that Canada and CHR have joined some of these big European projects. So Canadians are actually allowed to apply to these big projects, and this way not only get access to these collaborators, but also get access to some of these infrastructures and to these patient cohorts, because, well, there's at least tenfold more people over there than there are here, right? So therefore also tenfold more people with that particular rare disease that you're interested in.
I think becoming an international leader in the field is the first step.
No pressure. Well, thanks very much. Oh, go ahead, Nicole.
For sure.
Preclinical pipeline question. So I did a quick search. I saw that you had a DOC7 zebrafish model paper published back in 2010. So I'm just wondering, with your pipeline, did it start in the fish and then go to the mice and then on the patient? Was it nice like that?
Yeah, it's not always as it's in the diagrams, going from one thing to the next. It's sometimes jumping a step and then going back, etc. So I think some questions are just much quicker and much easier to answer in the zebrafish, and that's when we do it. Particularly if we have a new gene, like I showed you for the TFM gene, that we don't know much about and how it influences neuromuscular transmission, we very much like to go in the zebrafish first and get a quick first answer. And then if it's something more treatment related, preclinical, sort of setting up the scene for a clinical trial, then we often go into the mice if we have a suitable model. Mind you, sometimes the gene doesn't exist in the zebrafish, or it's there 3 times, so we can't use the zebrafish, and we use other models in collaboration, like fruit flies and worms and so on. So we're not very picky what model to use, but we like the zebrafish for sure.
Great. So thanks very much, Hans, for a wonderful grand rounds. Please fill out the grand round survey that's in the chat or will be emailed to you, and have a wonderful day.